Good afternoon. I am Diane Gage, a Marketing Manager at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled STR Profiling for Human Cell Line Authentication, presented by Dr. Brian Shapiro. Dr. Shapiro is a technical writer at ATCC. In this presentation, Dr. Shapiro will discuss many of the cell lines known to be misidentified and touch upon the loss and cure to the state of science by the use of these cells in research. In addition, Dr. Shapiro will highlight the tissue culture practices and testing procedures recommended by funding agencies, journal editors, and scientific societies to reduce cell line misidentification. Finally, Dr. Shapiro will focus on the use of short tandem repeat profiling for human cell line authentication. If you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. Any remaining questions, as well as the recorded webinar presentation, will be archived on the ATCC website, www.atcc.org. So with that, I would like to welcome Dr. Shapiro.
Thank you, Diane. I'd like to start with a brief word about ATCC. ATCC was founded in 1925 as a private, independent, nonprofit organization that serves and supports the scientific community with biological products and innovative solutions. We are a leading biological resource center and provider of biological standards and products encompassing a broad range of biological material, including continuous cell lines, bacteria, nucleic acids, and reagents. Over the past 50 years, cell lines derived from human cancers have played an important role in our understanding of the molecular pathophysiology of cancer and its treatment. During this period, we have seen a tremendous increase in the use of human cell lines as models for various cancers, and this has led to an increase in the use of cell culture activities and consequently an increase in cell cross-contamination and misidentification of cell lines. It is in this context that I will be talking about, first, the history of misidentified cell lines. How did we get here? Then I'll discuss how we can use STR profiling for human cell line authentication. I will then talk about some case studies. Then we will go into talking about steps for reducing cellular and microbial contamination. Finally, I will cover new regulations requiring cell line authentication. In 1952, HeLa was the first human immortalized cell line to be derived. This cell line came from the aggressive glandular cancer from a female of African origin, Henrietta Lacks. This cell line was derived in George Gay's laboratory by Mary Kubitschek, who was then at Johns Hopkins University. The cell line turned out to be the second cell line that was deposited in the cell biology collection at ATCC, CCL2. So HeLa was, as I mentioned, the first cell line to be isolated. But by the 1960s, multiple other human cell lines were also deposited. And what was found was that quite a few of these human cell lines, and other cell lines as a matter of fact, that were derived in the 60s were contaminated with HeLa. It was thought then that these contaminations were due to poor tissue culture environments. So in those days, it was not unusual for cell lines to be grown on the bench. There was no sterile plastic ware. There were no commercial basal media available. Technicians relied on a home brew of beef embryo extracts, human cord blood, and chick plasma. Attempts were made at creating an antiseptic environment by using Bunsen burners and steam for sterility. The technicians wore surgical masks, coats, gloves, booties, and hair covers. But these efforts were insufficient. HeLa's contribution at the time was that not only was it the first cell line, but it was also contaminating other cell lines. The contamination persisted and it became widespread. So what's going on? Is it that it was bad tissue culture collection, or was it just poor techniques? In modern days, we have a better environment. We have improved technologies, we have sterile plasticware and best practices into the culture, but contamination has still persisted. By 1967, several cell lines have been established. Misidentified cell lines went undetected because there were not good genetic markers to uniquely distinguish one cell line from another. Dr. Stanley Gartler, a geneticist from the University of Washington, was studying clonality. He looked at the segregation of the sex-linked glucose 6-phosphate dehydrogenase gene in various populations. What he observed was that the type A variant, or the first variant, was most often observed in individuals of African origin. To further studies, he ordered 20 cell lines from ATCC and found 18 of those 20 cell lines, or 90%, were contaminated with HeLa. Using isoenzymology, he observed that all of these cells were expressing, as I mentioned, the type A, or the first variant. Dr. Gartler presented his findings at the second biennial review conference on cell culture and organ culture at Bedford, Pennsylvania in 1962. The findings were, of course, not well received by the research culture community. The concern now was that contamination by HeLa was widespread in the tissue culture community and also at NIH. They asked Dr. Nelson Rees from the Naval Biological Laboratory to do a survey of the cell lines that were in use. He looked at 466 lines and found that 43, or 9.2%, had been misidentified. So now we're seeing widespread, not only interspecies contamination, but also intraspecies contamination. The technology that Dr. Nelson Rees used was cytogenetics and karyotyping, as he found HeLa markers in many human cell lines and other cell lines. So cell lines that were purportedly dog turned out to be human, monkey, or mouse. Cell lines that were supposed to be mouse or human turned out to be hamster. A cell line that was human turned out to be mongoose. A given cell line turned out to be human, and so forth. But the contamination, both interspecies and intraspecies, continued into the 80s and into the 2000s. In 1984, Huku and colleagues looked at 275 cell lines using karyotyping and found that 45% were misidentified. Using STR profiling, Hans Drexler from the German collection DSMZ looked at 252 cell lines and found that 18% were misidentified. He increased that number to 550 in 2003. Again, using STR profiling, he found that 15% were misidentified. Now, what's very interesting about this is that these are cell lines that were deposited in the German collection that came directly from the originators. Gertrude Buehring, in her landmark paper published in 2004, said less than 50% of researchers regularly verified the identities of their cell lines using standard methods such as DNA fingerprinting by short tandem repeat analysis. The contamination persisted. In 2004, we saw misidentification of breast and prostate cell lines. In 2007, again, breast cell lines. In 2008, thyroid cancer cell lines. In 2009, adenoid cystic carcinoma cell lines. In 2010, esophageal adenocarcinoma cell line. In 2013, thyroid and lymphoma cell lines. So we can see that the misidentification of cell lines is widespread and has affected a wide variety of cancer tissues. In a landmark paper published by Boonstra and his colleagues from the Netherlands, they looked at 40 esophageal adenocarcinoma cell lines and found that 3 of the most commonly used cell lines were misidentified. SEG1 turned out to be a lung carcinoma. Big one turned out to be a colorectal adenocarcinoma. SKGT2 turned out to be a gastric fundus carcinoma. Now, what's very interesting about the study is that the authors showed that over 100 scientific publications had resulted from these misidentified cell lines. There were at least 3 NIH cancer research grants and 11 U.S. patents, and they'd gone on to recruit patients for clinical trials. So it is now estimated that 1/3 of all cell lines in the public domain used in the last 50 years, scientists have misidentified. And the majority of these cell lines that were used especially for preclinical research are human in origin. If you look at the ICLAC database of misidentified cell lines, that's the International Cell Line Authentication Committee database, you'll see that there are about 438 misidentified cell lines, of which 423 are of human origin. So what are some of the consequences of the misidentified cell lines? Well, among them, the loss of a cell line, the loss of time and money, misinformation in the public domain, discordant or irreproducible results, and a tarnished reputation. Jeffrey Boatwright from Emory University stated in the most recent article that was published in the Scientist magazine on the Big Cleanup, stated, "If we're not using what we think we're using, we're not testing our hypothesis. We're just gumming up the literature. I'm not sure what we're doing, but that's not science." So we do highly recommend that you routinely test your cell lines with short tandem repeat DNA analysis, or STR DNA analysis, for intraspecies identification. So what are short tandem repeats? STR are microsatellite DNA that are randomly scattered throughout the genome and are among some of the most informative markers for human identity. Each repeating unit within this tandem repetitive sequence is about 1 to 6 base pairs. There are multiple loci across the genome, and the degree of repetition is usually about 5 to 100. So again, we highly recommend STR profiling to uniquely identify human cell lines. A bit more about STR analysis for human cell line identity. There are certain features of using STR profiling. So the target sequence consists of microsatellite DNA, as I mentioned. It typically needs about 1 to 2 nanograms of DNA to perform the analysis. There's typically 1 to 2 fragments, 1 in the case of homozygous and 2 for heterozygous, and I'll be talking about that a bit later. There are discrete alleles that allow for a digital record of data. In other words, each allele can be converted into a numerical value, and the database can then be developed from that. The markers are distributed throughout the genome and are highly variable within the human population, and consequently are very informative. So here we have an example of 8 STR markers. And as you can see, they are distributed in different chromosomes. For example, D16S539 is on chromosome 16. The repetitive motif is GATA. There are 5 to 15 number of repeating units, and there are 10 alleles associated with this locus. In the next locus, D7S820 is located on chromosome 7 on the QR long arm of chromosome 7. And again, you can see the number of repeating units and the number of alleles. These 8 markers are the minimum number of markers that are recommended for STR analysis that can uniquely identify a human cell line. This was actually based on the retrospective status of 500 human cell lines. Now, in each of the kits that are available commercially, amelogenin is also included. This is not a short tandem repeat, but is used for gender determination, and it can be used to determine whether the cell line was derived from a male or a female. If you were to use all of these 8 STR markers in a multiplex reaction, the power of discrimination, or the probability of 2 lines having an identical profile, is 1.21 to 2 times 10 to the 8th power. So what are some of the characteristics of STR markers? High discriminating power, high observed heterozygosity, robust and reproducible results can be obtained, low set of characteristics, and a low mutation rate. The advantages of STR analysis include the banding pattern is reproducible, it's PCRable, and so high throughput analysis is possible. Small fragment size allows for multiplexing. There's an internal allelic ladder that simplifies interpretation, and I'll talk a little bit about that later. The small product size is compatible with partially degraded DNA. Rapid processing is attainable. The probe is human-specific, and highest stringency conditions are applied to the PCR reaction. So this is a schematic outline of the procedure. You can start with either purified DNA in the liquid form, or alternatively, you can spot your cells onto a FTA filter paper. The cells, when they come in contact with the paper, will lyse, and the DNA will bind to the paper. You can then punch the filter paper and put that into your tube for the PCR reaction. So you don't have to do DNA purification. After you've performed your PCR reaction, the amplicants are then separated by capillary electrophoresis in the presence of internal lane standard and the allelic ladders. For data analysis, you'll get a readout electropherogram after you've converted the fragments into different alleles. Each allele will have a numerical value, and that numerical value you're now able to convert into a database. The requirements for performing STR analysis include you'll need a gene sequencer, a thermocycler, a commercial STR kit, and all kits have amelogenin, as I mentioned. You will need an STR database to confirm or verify that you have a unique profile. And of course, you need experienced technicians. The experienced technicians are not only needed for the running of the assay, but probably just as importantly or even more so for the analysis of the data. So as we talk about DNA polymorphism, and we're talking about the informativeness of these markers, if you look at the upper left panel in this example, we have individual A, who is homozygous for the locus D16S539. As I mentioned, this marker is on the long arm of chromosome 16. And in this individual, if you're looking just at this locus, you will have 8 repeating units that came from the maternal chromosome and 8 repeating units that came from the paternal chromosome. So if you do the PCR reaction followed by the electrophoresis and do the analysis, you will get one band. You will get a co-migration of the 8 repeating units on maternal chromosome and the paternal chromosome, and they'll co-migrate, as I mentioned. It is assigned the numeric value of 8 in accordance with the allelic ladder. Now for the second individual, that individual would have received 10 repeating units from the maternal chromosome and 9 repeating units from the paternal chromosome. When you would have carried out the assay, you can get 2 bands, and this is heterozygous because you're getting one that corresponds to the 9 repeating units and one band that corresponds to the 10 repeating units. If you have different other individuals, as in the bottom panel shows, in the case of Tom, he has 9 repeating and 13 repeating units. So that's 9 to 13. Dick is homozygous for 10, and so that would be a 10. And in Harry, it would be heterozygous for a 9-12. So you can begin to see where you're now getting unique profile for each individual based on the number of alleles that is present for each marker. Information can be even more robust by adding additional markers. And here we have an example of 8 STR markers. If you look at D5S818, the frequency of that allele from the maternal chromosome is 1 to 10, as is the frequency from the paternal chromosome, 1 to 10. And the probability, just using that one marker of 2 cell lines, having an identical profile is 1 to 20. If you do the frequency or the probability of all the loci, of all 6 STR loci, then you're multiplying the product of all of these probabilities. So the number you would be 1 to 9.3 times 10 to the 7th power. So the more markers you add, the more resolution you have, and the higher the probability of a uniquely identified cell line. Gender is also very important in cell line identification. As I mentioned, all the commercial kits have an amelogenin gene. The amelogenin X gene is on the X chromosome, and the Y gene is on the male chromosome. The difference between the male and the female is that there's a 6 base pair dilution in the first intron. So in the male, you will have 2 bands, one that corresponds to amelogenin X and amelogenin Y. And in the female, you have 2 X's, so they co-migrate together and you get only 1 band. So this is an example of a readout or an electropherogram of 2 unrelated cell lines. We're looking at 8 markers plus amelogenin. In the first upper panel, we have K562 cells, which is a chronic myelogenous leukemic cell line. You can see that the fragment sizes have been converted to a numerical value or alleles as displayed in the table below. The WS1 is a fibroblast cell line, and it is different from the related individual from K562, and it's a female. If you look at the numerical value of the K562 compared to that of the dubious one, we can see that they are unique. So K562, 2 unrelated individuals, unique STR profile. Compare this to these 2 related cell lines. These are 2 cell lines derived from the same individual. We have HAAE2, which is from an endothelial cell line from the aorta. We have HFAE2, which is from the femoral arch of the same individual. Both are endothelial cells, but if you look at the STR profile, they're identical as you would expect. STR profiling can also link a clone to a parental cell line. Here we have BGO1, a human stem cell line. If you look at the STR profile of BGO1 to the clone, which here is BGO1V, you will notice that it has an identical STR profile. However, it's very important to note that the clone could be functioning or acting differently from the parental. And in this example, that's the case. The BGO1, the parental, has normal diploid chromosome. In the case of BGO1V, which the clone is a clone of the parental, it's aneuploid. It has 3 copies of chromosome 12, 3 copies of chromosome 17, and an additional copy of chromosome X. In this case, the BGO1V at the phenotypical or at the STR level anyway is identical but could be functioning differently from the clone. There's data that shows indeed that the BGO1V actually grows much faster than the parental cell line. Now you can also use STR profiling to monitor the stability of your different cell banks, like we do here at ATCC. When we get a cell line in for the first time, we want to see what the profile is of the donor material. So we open that vial and from that donor material. We spot it onto an FTA card and then perform the STR analysis and get a baseline profile. When we expand that culture to make our pre-master cell bank, again, we repeat the STR analysis and compare it to the donor material to see whether or not there's any change in the profile. Not only are we looking for change in the STR profile, but also just to make sure that there is no cross-contamination from the stock. We will also then take one of those vials of the token, expand it, and make a seed or master cell bank, and again repeat the STR profile, making sure that there's no changes and no contamination. And when also take one seed stock and expand it to make a distribution or working cell bank, we repeat the STR profiling. So we want to make sure that the STR profile is consistent from the donor material to the working cell bank. But just as important, we make sure that there isn't cross-contamination. STR can indeed pick up cross-contamination for other cell lines at a level of about 10%. So here's some common sources of cellular contamination. Getting cell lines from your colleague down the hall. Continuous culturing of working cell banks. It's very important that when you get a cell line in, whether it's from us or from other sources, is that you put down your own seed stock. The cells will change over time. Most kept cells will change in culture, that is to say. And so it's very important for you to freeze down some of your seed stock. But also by freezing down, you're preventing the risk of absolute cross-contamination and just the total ruining of your cell line. The use of feeder cells can also contribute to cell contamination. Mislabeling of culture flasks, working on multiple cell lines concurrently can also induce cell contamination. Using just one reservoir of growth medium for multiple cell lines can also induce cell contamination. And so it's very, very important that at any time you're in the process that you really know what cell line you're working with. So in this case study, this shows an example of the cross-contamination at its early stage. We have SKOV3, which is an ovarian cell line that was contaminated with another cell line indicated in yellow. We notice contamination because we have multiple loci, multiple sites. And so this is really an early stage of contaminating cells. It's not always easy to catch these unless you're performing routine STR characterization. Usually, the contaminating cells will take over the culture in a very short time. And so testing your cells routinely is very, very important. Here, we have an example of gender misidentification. There was a cell line that came to us and it was purported to be female in origin. When we performed the STR analysis, we found Y material. We were able later to confirm that this Y material was real by using whole chromosome paint to highlight the Y chromosome. In this third case study on misidentified cell lines, this was a cell line X that came to us. We performed the STR analysis. We compared the data to our database and found that the cell line was identical to HT29, which is a human colon adenocarcinoma cell line. We looked at the morphology of the cells, and we can tell that even the morphology of the cell line X is similar to that of H29. We had access to the karyotyping that was done previously, and we compared it to that of H29 and found that they had common chromosomal markers. In the fourth case study on microsatellite instability, as I mentioned before, what we would have looked at our donor material and performed STR analysis and compared to all subsequent stocks of cells, the working cell bank and the master cell bank. We're also looking for instability of some of the markers. And in this example, in D13 S317 at allele 9, we have seen instability in the seed and it disappeared at the first, or it was reduced for the distribution stock. Then, by the second distribution stock, it disappeared and reappeared in the third distribution stock. So again, how you culture your cells, how you manipulate the cells could have an effect on instability. This could also be an inherent feature of this particular locus. So in 2012, we actually published a standard, a consensus standard, ASN 0002, of the authentication of human cell lines, standardization of STR profiling. And this consensus standard, which is a written standard, we want to be sure that the standard was described consistent, inexpensive, and universally applicable method for authenticating human cell lines. And this method is used widely by many of the testing services. The group that made up the working group were internationally known experts within the field of STR profiling. So some steps for reducing cell contamination. Always use good documentation. Highly train your technicians, especially in good aseptic techniques. Do not use one reservoir of medium for all of your cell lines. So be sure you use one reservoir per cell line. This cannot be overstated. If you're working with multiple lines that are using the same medium, you must aliquot and use a reservoir of medium per cell line. Also, be sure and aliquot your stock solutions and reagents. Label your flasks legibly with the name of the cell line, passage number, and date of transfer. And if possible, use barcodes or a LIMS system whenever available. Work with one cell line at a time in the biological safety cabinet. Clean your biological safety cabinet between each cell line and allow a minimum of 15 minutes between each cell line, between working with each cell line, that is. Quarantine dirty cell lines from clean cell lines. And what I mean by this is that if you bring a cell line into your laboratory for the first time and you have not tested it to see if it is the correct species, free of microbial contamination, especially mycoplasma, then you need to separate these cell lines until they've been tested and found to be the right species, free of microbial contamination, including mycoplasma and fungi, and just making sure that they are correct by short tandem repeat profiling. If you don't have the physical space in which to do this, what you may want to do is wait before you use the dirty cell line. Wait until the end of the day. Make sure that the biological safety cabinet has been cleaned before and after use again. Some other things that you can do, by the way, is make sure you have a manageable workload. That will reduce accidents, cross-contamination. It's a best practice to do your culture in the morning rather than later in the day when you're tired. Try not to do your cell culture late on Friday when you're rushing. That's when you're most likely to have accidents and cause cross-contamination. A clean laboratory will reduce bioburden and prevent microbial contamination. Legible handwriting is also very useful in preventing cross-contamination. In addition, monitor your cell lines, monitor them routinely, create a good working environment, and also review and approve laboratory notebooks and obtain cell lines from a reputable source. In our citation for cell authentication, looking at STR profiling and using the short tandem repeat and STR profile, you can see, looking at Highwire Press citation over the years, there's an increase in the number of those citations. It seems to imply that journals are now requiring scientists to authenticate their cells, and people are responding to this. So here's just a partial list of the journals that are now either requiring or recommending cell line authentication as a prerequisite for publication. NIH has recently revised guidelines to applicants for funding under enhanced reproducibility through rigor and transparency. This became effective on January 25th, 2016. In this guideline, they recommend authentication of key biological and/or chemical resources. NIH expects that key biologicals, including cell lines, will be regularly authenticated to ensure their identity and validity. NIH also published principles and guidelines for reporting preclinical research. This is a 5-point guideline. In number 5, they talked about considering establishing best practice guidelines for cell lines. And under cell lines, they recommended reporting the source of the cell line, authenticating the cell line, and the status of mycoplasma contamination. Over 130 journals, scientific associations, and scientific societies have signed on. Also, the Prostate Cancer Foundation has issued a cell line authentication initiative. In this initiative, they are also recommending genetic and pathogen testing of cell lines for future funding. In Nature journals, authors are asked to report the source of a cell line to indicate whether the cell line is authenticated and to check the cell line against a database of misidentified cell lines such as the ICLAC database. In an article recently published in PLOS Biology on economic reproducibility in clinical research, they did a study of the NIH report of projects using cell lines or cell culture and showed that over $3.7 billion have been spent on such types of research using cell lines or cell culture. They argue that if you take into consideration that 10% of cell lines have been misidentified, then over $370 million would have been wasted. That article went on further to say that if you look at the average funding of a 4-year grant over a 4-year period, which runs about $450,000, and you test one cell line at about $200 per test, And so if you spend about $1,000 just doing STR analysis to authenticate your cell lines, then that only represents about 0.2% of your total research budget. So testing cell lines is very, very important. The consequences of not doing is even far-reaching, and it's really not that expensive to do. So when should you perform STR profiling? Well, when you first receive a cell line into the laboratory from an unreliable source, after 10 passages, after preparing a new cell bank, and whenever in doubt. Testing services for STR profiling of human cells. There are several testing services, including ATCC and other cell banks, paternity testing laboratories, universities, and collabs. But most importantly, a consideration in choosing a testing service is that the technicians are highly trained and experienced, especially in the interpretation of the data, and that there is access to a database on STR profile of human cell lines. Thank you again for your attention, and I'm ready to take your questions.
Thank you so much, Dr. Shapiro. That was very interesting. In just a few moments, we will begin our Q&A session. Please use the chat function available through the webinar program to submit questions. This session will be documented as a PDF and archived along with the recorded webinar presentation on the ATCC website at www.atcc.org. First question. Are there other methods to authenticate cell lines besides short tandem repeat STR profiling?
Yeah, good question. So in addition to STR profiling, there are a number of ways to resolve the identity and status of a cell line, including growth curve analysis, cytochrome C oxidase 1 barcode assay, that's for species identification, karyotyping, random and amplified polymorphic DNA analysis, and SNP analysis.
Great. So another question is, what is the easiest way to submit a human cell line sample to ATCC for STR DNA profiling?
Sure. So really the easiest way is just to order the service through ATCC. Once you've ordered the service, we'll send you a sample collection kit in the mail. So once you receive this kit, you'll just spot your cell suspension directly onto the FDA paper, dry it in your biosafety cabinet, and then mail it back to ATCC in the pre-addressed envelope. The collection kit will include the barcoded FDA paper, sample prep instructions, the sample submission form, some desiccant, and the envelope for return mail. And so the reports will be emailed to you by the ATCC within 5 business days of order confirmation and receipt.
Awesome. Let's see. Considering the extent of cell line contamination present in many labs around the world, what cells were used to develop ATCC's STR profiles?
Okay, so ATCC has consistently authenticated our collection of continuous human cell lines through STR profiling for many, many years. All of our cell lines are included in our database along with cells that have been de-accessioned due to misidentification. So HeLa is really the primary culprit for misidentification and cross-contamination of numerous other cell lines. We actually have a lot of information about cell authentication and contamination. They can be found under our Cell Biology Resources webpage, and also in the November 2014 article, The Dirty Little Secret of Cancer Research, which is in Discover magazine. And it does a nice job of covering the history and scope of cell line contamination.
Great, great. Oh, here's an interesting one. What is the repeat slippage? What is its cause?
So that is a good question. Repeat slippage is also known as stutter. A stutter is a single repeat unit smaller than the true peak, which can occur immediately before the true peak. These peaks, which may result in differences between two samples of a cell bank at one locus, are typically less than 15% of the true peak. So the stutter is caused by errors in DNA polymerase-mediated amplification.
What threshold of match probability is utilized for determining identity?
Okay, so using the masters or alternative masters algorithms, cell lines with greater than or equal to 80% match are generally considered to be related, meaning they're derived from the same patient or donor. Cell lines with a match ranging from 55% to 80% require further profiling for authentication of relatedness.
Gotcha. Now, I know you mentioned this in the deck, but how often should I authenticate my cell lines?
Sure, I'm happy to repeat actually. When you first receive a cell line into the laboratory from an unreliable source, after 10 passages, after preparing a cell bank, and when in doubt, check your cells before you wreck yourselves.
That's a great one. Love it. And is there a recommended maximum passage number for a cell line before it's no longer useful?
That's a good question. That's a really good question. The limit to the number of times a given cell line should be passaged is pretty much determined empirically for each cell line based on the nature of the cell line and the type of work you're doing with it. So although many immortalized cell lines have an indefinite life expectancy, the characteristics and properties can change even markedly when animal cells are cultivated for extended periods in vitro. So we work to maintain the cells at the lowest passage possible using our seed stock system, which we definitely recommend every laboratory use. As a general rule, it's best to passage the cells a limited number of times before returning to a lower passage frozen stock to ensure the functionality and characteristics of the cells are maintained. We recommend passaging a culture no more than 8 to 10 times or 2 months. It is the short answer to all that.
That's great. That's great. Now, I understand from the presentation that STR profiling is currently only available for human cell lines. Do you have anything for animal cell line authentication?
Yeah, so we're working to validate primer sets for mouse cell line STR profiling. We have a CO1 assay that can be used for interspecies identity. So the CO1 gene region that I'm talking about, it's used as a standard barcode for almost all animal groups. It's a 650 base pair region in the 5' region of the mitochondrial cytochrome C oxidase 1 gene. And it can be used to discriminate between species of the same genus.
Great. Great. And I have one here from someone. They work at a small university with few resources. However, they're not able to do cell culture. What's the most basic, cost-effective way for them to identify cell lines when publishing?
Sure. So, of course, I'm going to say submit your sample of your cells to a testing laboratory like ATCC. Some other testing laboratories that can provide STR profiling are cell banks, paternity testing labs, universities, core labs. But I guess I can't stress this enough. When choosing a testing service, it's important to consider the training and experience of the technicians who are performing the service, and that you have access to an STR profile database to compare your cell line profile against another for a match.
Okay, well, thank you so much. And at this time, we'll conclude our Q&A session. I would like to thank Dr. Shapiro for the excellent presentation, and thank you everyone for attending this webinar today. Any questions that were not answered this afternoon will be answered and posted with the video at www.atcc.org. Please join us for more webinars in the ATCC Excellence in Research webinar series. Thank you again for your time today, and have a great day, everyone.